I will talk about work that I have done at the University of, basically from my doctoral work and beyond. So the title of my presentation today will be on the behavioral, computational, and motor neuroscience. So what we can learn from the basic mechanisms that we can apply to clinical applications. So today I will talk a little bit about my training background. Then I will identify who I am as a researcher, what my research interests are. And then I will talk about my work at the University of Delaware, which was basically the behavioral part of the presentation. Then I will talk about the ongoing work here at the University of Nicosia, which is the computational component. And also in the end, I will talk about the motor neuroscience, which was combination of my doctoral work, my second postdoctoral work, but this project that I will present today, I will run it hopefully in the near future here at University of Nicosia. So in 2001, before 9/11, 3 weeks before 9/11, I arrived to Los Angeles, California. I earned 3 degrees in 10 years. As Dr. Hadjipapas says, I started with exercise science, then I moved to clinical biomechanics, and then more theoretical on motor control and learning. Then I drove 4 days cross country, and I went to Charleston, South Carolina, where I spent the next 4.5 years doing my PhD there in health rehabilitation science. Then I spent then I did my first postdoctoral work at the University of Delaware, and then I moved to Manhattan, New York, doing my second postdoctoral training in neurology at New York University School of Medicine. And then since December 2019, I started my third postdoctoral training in neuroscience with Dr. Hadjipapas. So my identity as a researcher is, given my background, both degrees and postdoctoral training, basically identify myself as a motor neuroscientist with focus on impaired movement after neurological injury. I'm particularly interested in stroke. So my research interests basically are neurophysiology of the motor descending pathways, which that I will talk at the end of my presentation, the neuromechanical control of locomotion, meaning walking, and the goal-directed stepping actions. Also, I'm very interested about neuroplasticity, motor recovery, and rehab after stroke, meaning how people who had a stroke can get better in terms of movement. And lastly, I'm really interested to apply computational approaches, which I'm learning here, working with Dr. Hadjipapas, how to quantify neurophysiological and neuromechanical measures. By doing that, can improve the way that we assess interventions in clinical cohorts. So before I started with the research, let's define the 3 types of neuroscience that I will present research on today. So behavioral is basically neuroscience is the application of the principles of biology to the study of physiological, genetic, and developmental mechanisms of behavior in humans and other animals. In my case, I'm interested about the behavior during walking and goal-directed steps, both in humans, both in healthy and clinical cohorts, but also in terms of science, I'm very interested in macaque monkeys. Computational neuroscience basically employs mathematical models, theoretical analysis, and abstractions of the brain to understand the main principles that govern the nervous system. So basically, here, what I'm interested is how we can use these models to tell us something about the motor system. And motor neuroscience basically focuses on the regulation and control of muscles, therefore movement. I'm particularly interested in how the brain controls specific movements and how the movement can be restored after a brain injury. So we'll start with the behavioral neuroscience, basically the work done at University of Delaware. So the main theme of those three studies that we'll present to you is the effect of priming on locomotor learning. So let's define, first of all, what is priming. So it's originated from psychology, so it's defined as a change in behavior based on previous stimuli. So it's a type of implicit learning, so we don't know that we learn about it. So the main action of priming is basically primes gets ready the brain. So therefore the brain is more responsive to accompanying or subsequent training, for example, motor. So basically the motor priming is the repetitive movement that is not goal directed. So it's not skill based. So there are different paradigms, but the one that I will present in the next few slides is the aerobic exercise, and particularly the high intensity exercise. So we know that all of us, we know about the indirect effects of exercise. So we know that when we exercise, we get some improvements in general health, but also there are some direct effects of aerobic exercise on the molecular signaling pathways in the brain, meaning that can increase neurotrophic factors such as BDNF, brain-derived neurotrophic factor, which can increase, can promote or boost neuroplasticity, neurogenesis, and neuroprotection, and then that can boost cognitive function, for example, learning, memory, and attention. So therefore, aerobic exercise, in addition to the general improvements in health, can also have some direct effects on learning and memory and attention. In my collaborators in Delaware were very interested in learning and memory of movement. So there was quite a few studies. So this study started around 2015, 2016. So around that time, there were enough evidence which showed that exercise can boost motor learning, and the mediator is BDNF. So the two parameters of exercise that can boost motor learning is basically the intensity and timing, meaning the higher the intensity is, which has been quantified with the lactate changes, can improve motor learning. Also, it's important when the exercise is administered relative to the motor task. Let's say you want to learn how to play tennis. When do you have this type of priming? Same day, 2 days later, right before the task, right after. So at the time, evidence showed that it should be the right before or right after the motor task. So before we move to the actual studies, let's introduce you what locomotor learning is. So in this particular paradigm, we define locomotor learning as the sensorimotor adaptation. So let me show you a cool experiment. So as you can see here, so somebody works on the treadmill, which has, as you can see, two belts. So each belt can go to different speeds. So here, what we call baseline, both belts are moving at the same speed. So as you can see, the step length is very symmetric between the right and the left leg. Then, so that's what this dots indicates. So straight line meaning symmetric gait pattern, which was quantified with a step length. Then we introduce the perturbation, meaning that one belt goes as double in speed as compared to the other. So in this case, check the left leg will go to 0.75 millimeters per second, whereas the right leg will go 1.5. So look how asymmetric the gait pattern will be. And this video was taken at the beginning of the adaptation. So here we have a 15 minutes of data. So see how asymmetric the gait pattern is. The person is perturbed, and at the beginning of this motor task, as you can see, the person is very symmetric. But what is interesting about the nervous system is what the humans and also animals can learn how to deal with this perturbation. And as you can see, within few minutes, people can walk symmetrically, even the treadmill still runs at two different speeds. Then immediately after, we want to see how much that person learned. So we do what we call de-adaptation. So now both speeds, both belts are moving in the same speed. As you can see now will be the reverse phenomenon. Now the asymmetry is reversed. So how fast this data goes back to the baseline that shows how fast the person can de-adapt. Okay, now how we quantify now the locomotor learning. So we like to see how much the person has learned during the session, but also when the person comes the next day and is presented with the same task, how much that person retains in terms of this type of learning. So as you can see, adaptation is the difference between the beginning of the task compared to the end. So the bigger the magnitude is, the bigger the adaptation. And then when the person comes on the next day, we like to see where the person starts. So the bigger the difference is between day one beginning and day two beginning, that shows that the person retained a motor memory of this task. So the greater this, that means better retention. And also, as you can see, most of the times people go back to the baseline symmetry. But remember that the treadmill still moves at two different speeds. So my group, it's the group that I work at Delaware, is well known about the locomotor learning and rehabilitation in people post-stroke. So we know that stroke is one of the leading causes of long-term disability, and walking is one of the impairments after the stroke. So relearning or learning to walk is the main goal in stroke rehab. So learning of a novel walking task, in this case, the split belt treadmill walking, can occur after stroke. So there are quite a few studies from that group that show that people can learn how to walk. Stroke patients can learn how to walk on a split belt treadmill. Yet the rate of learning was slow compared to normal. So for this series of studies, we wanted to ask the fundamental question: how can slow locomotor learning be improved after stroke? And given the evidence I showed you before about priming, we decided to see the effect of exercise on locomotor learning. So we ran 3 studies. The first one, we ran it in order to examine whether an acute high-intensity exercise bout would result in significant exercise-induced responses in people post-stroke. Basically, we want to see whether this priming paradigm, which was the first at the time, is feasible for people with stroke. Then we ran 2 more studies that basically we examined whether this type of priming would increase locomotor learning in people post-stroke, but also healthy controls. So we ran the study with stroke first and then the healthy. So study 1, basically the question was, is an acute high-intensity exercise bout feasible in chronic stroke patients? So so the goal was to exercise continuously for 5 minutes at a target intensity. So our control group was basically low intensity, and we have 2 high intensity groups. The first one was the TMW, which in basically subjects walk on a treadmill at their fastest comfortable speed, whereas the second high intensity exercise group was the total body exercise, basically moving both arms and legs, depending which one they could, the stroke patients. in order to reach certain levels of heart rate maximum, but also lactate. So what we found that basically both groups, both high intensity exercise group, they exercise at the target intensity. They spent most of their time, those 5 minutes they spent at that target intensity, which basically shows that they were able to work out at that, to reach that target intensity, but also to work out at that high intensity by showing changes in lactate, but not in BDNF, which, as I showed you before, BDNF is the mediator between exercise and motor learning. So what is interesting is also that the TBE had some differences with the TMW, basically the total body exercise with the treadmill walking, and that was basically people in that group that were able to reach higher intensities. They have created changes in lactate. So basically, we showed that this type of exercise was feasible and had an effect only on lactate measures in chronic stroke patients. Then we said, okay, given that now the priming is feasible in stroke patients, we collected data on 37 people post-stroke, and we wanted to see whether a single bout of high intensity exercise can increase the learning of that specific locomotor task in people post-stroke. So as you can see here, people came in 2 sessions. In the first session, there was 1 minute baseline. Basically, we wanted to assess their baseline symmetry in terms of step length. So we manipulate both the timing and the exercise intensity, meaning the control and the treadmill working. The exercise was administered right before the task, whereas the total body exercise group, the exercise was administered immediately after the task. Okay, and then all participants came 24 hours later, and we just ran the locomotor task. Basically, we wanted to see, okay, how much they remember from day 1 locomotor task. So the way that we quantify the locomotor learning, again, we look between the days and within the session magnitudes, but also we look the rates, meaning that the faster the rate is, the fastest the learning was. So as in study 1, we found that we found similar results in terms of the exercise-related measures. Both high-intensity exercise groups, they worked out at specific intensities and a high percentage of duration during the exercise. Also, there was increase in lactate, but no change in in BDNF. So then we look the locomotor learning task. So let me orient you in this figure. So again, 0 basically is symmetric gait pattern, and the farther you move from the 0, the more asymmetric the pattern is. So remember that the control and the treadmill walking group, the exercise was before the task, whereas the total body exercise was immediately after, and that the treadmill and the total body exercise groups were the high intensity groups. So what I'm plotting here is the trial to trial step length asymmetry of 15 minutes of working on day 1 and 15 minutes of working on day 2. So just by looking the lines, which is basically thick lines for each color is the group mean, and the shaded is the standard deviation, the variability, you will see that there are no differences between the 3 groups. So neither exercise intensity nor exercise timing had an effect on locomotor learning for both adaptation and retention in people post-stroke. We say, okay, at the time that this study was done, there was more evidence. Again, all those studies that I showed you before was in healthy adults. The exercise was basically bike, high-intensity bike exercise, and the task was goal-directed upper extremity. Either just move your finger and match the trace that you are looking in the computer, or just match force production by squeezing a force transducer with your arm. And all those studies were in healthy. So by the time this study was done, there was evidence showing that, okay, the best time to have the priming is right after the motor task. And the scientific rationale of that was because it's the beginning of the consolidation. It's the time that the brain is more prone to learn. And then also the other piece of evidence, it was that the robust effect of priming on motor learning was manifested 7 days after the motor task. So we designed study 3, just healthy adults. We have 2 groups. That the first group was just rest and the other group was exercise. So was healthy adults. We have them come 3 days. Day 1, that was basically the introduction to the locomotor task. So all these subjects never worked in the split bed treadmill paradigm. Then they came for session 2 and session 3. So on session 1, healthy subjects at rest, they just sat down, whereas the people at the exercise group, they did the total body exercise. They did the total body exercise, which was shown in the previous study that has the biggest effects on exercise-related measures. But we increase the high intensity, so healthy subjects exercise at 73 to 95% of the maximum heart rate. This very high intensity. But we also wanted to see changes of lactate more than 10 millimoles. As you remember from the previous table, in stroke, the maximum that we got was around 6. So we wanted to replicate what previous work done in upper extremities. And also that we quantify retention on session 2, but on day 1 day after the session 1, but a week later. So here is also the stride to stride step length asymmetry. It's just reversed, excuse me. So the red line is the exercise group, whereas the blue line is the rest. Given that the exercise was done after the locomotor task on day 1, we expected these lines to be very similar, and they are. Now, what we expected is to see differences between the red and the blue lines on session 2 and session 3. We expected that the red line should start closer to the 0 line, and also the slope to be more abrupt, meaning better rate. And that should be also the same for session 3, meaning 1 week after the day 1 that the locomotor task was introduced to the subjects. As you can see, there were no significant difference in any locomotor learning measures. So again, as in stroke patients, a single bout of high intensity exercise, even though we tried to replicate what other studies did in upper extremities, had no effect on lower extremity tasks in healthy controls. So what we learned from those 3 studies? So that first of all, an acute short high intensity exercise bout be feasibly implemented in stroke rehabilitation and can be coupled with locomotor learning task. However, may not be an effective primer for improving adaptation-based locomotor learning in either stroke patients or healthy controls. Now, the benefits of exercise effect may depend on the type of motor learning. So the previous work was basically skill learning, whereas in our studies, the type of learning was sensory motor adaptation. Also, keep in mind that these 2 types of motor learning are controlled from different parts of the brain. And also, the task may have an effect on these results, meaning that in the previous work, the task was arm accuracy tracking task, whereas here we had the people work on the treadmill. So that was my work for my first postdoc in Delaware. So that's what we just finished, the behavioral neuroscience part of the presentation. So after 18 and a half years in the United States, I decided to come back in Cyprus. I was fortunate. I was accepted to do a postdoctoral fellowship with Dr. Hadjipapas in computational neuroscience. So what I will show you now is going to be the ongoing work that Dr. Hadjipapas and I are working on with collaboration with scientists from Netherlands and United States. And the title of this the project that I will show you results is the cortical electrophysiology of the visual cortex in non-human primate. So we're moving away from behavior, we're moving from the motor cortex, we're relocating to the visual cortex, and we're moving from humans and we're going to macaque monkeys. So there is a lot of work in neuroscience, there is a lot of work on micro scale and macro scale analysis. So the macro scale is the subcellular and cellular and molecular level, whereas on the other side of the spectrum, there is a lot of work about the areas, the systems, and the global networks. But what is still unclear is the mesoscale analysis, meaning that how the local networks in the brain communicate, how the cortical layers and columns communicate with each other, and how and what factors can influence those layers, and how what we can learn from those basic mechanisms that we can apply in clinical cohorts. So I will focus for the next few slides on the cortical layers. So in the left panel here, you can see the difference. So it's the neocortex, meaning the gray matter around the brain, and you can see that all of them, they have the same number of layers, but the thickness of the cortical layer differs depending the part of the brain. So we're interested in this study on the primary visual cortex. And as you can see, layer 4 is one of the thickest, and it's because the prominent sensory input from the thalamus. So the visual cortex is responsible for what we see and how we process visual feedback. Visual input. So one of the methods that we can quantify what is going on in those cortical layers is by collecting local field potentials, LFPs, but also spikes for multi-unit activity. So in the paradigm that we use for this particular project, basically this was done in Netherlands when Dr. Hadjipapas was working there. So pretty much we implanted 2 laminar probes in monkey's brain in 2 visual cortices, V1 and V4. So V1 is the primary visual cortex. So as you can see here, so the sensory input goes through the eyes, through the nerves, and then goes to the V1. Then goes to V2 and then V4. So we have 1 laminar probe at V1 and 1 laminar probe at V4. As you can see, the laminar probe has 16 recorded contacts. So that allows us to collect activity at all different layers of each visual cortical area. So here is the paradigm. So pretty much we have 2 monkeys. Each monkey had to do 2 conditions and in multiple sessions. So pretty much the experiment was, so there was 2 attention conditions. The first condition, the monkey had to fixate their eyes to a stimulus, which the stimulus was the target here. which basically was manipulated in terms of orientation, either 45 degrees or 0 degrees. And also we manipulated the contrast. So the contrast meaning that either blurred target or very visible. By doing that, we're able to manipulate the amount of sensory input in the visual cortex. So the goal here for this attention, for this condition of attention, was to report the orientation. Whereas the second attention condition, the monkey was learn how to ignore the grading and basically just to report the color change at fixation. What I forgot to mention that before any experiments, the monkey learned to move the eye towards a direction depending on the orientation of the target. That was in there for attention 1, whereas in attention 2, the stimulus was irrelevant. So this analysis is ongoing. Even though we're interested how the cortical layers are communicated with each other, for now, for this presentation, I will show you only the effects of the contrast, orientation, and attention by collapsing all sessions together and all layers together. So this column is about V1, this column is V4. First row is the contrast, orientation, and attention. So in both visual cortices, as you can see in terms of contrast, as the contrast increased, the response became bigger and faster, meaning that as more visible the target was, the response in the visual cortex, quantified by the firing rate, meaning how many spikes per second, got bigger but also faster. Orientation, as you can see, there was no effect in neither cortex. Also, attention has an effect on the firing rate, and we can see that the blue line, which is the attention 1, basically the monkey had to attend, was bigger than the attention 2, which the monkey was learned to ignore the grading. Another piece of information from this figure is that, as you remember from the 2 previous slides, the sensory input goes first to V1 and then to V4. So as you can see that the gray squares is basically the range of the peak onset. You will see that the peak onset for all 3 factors was faster in V1 compared to V4. Last piece of information about this figure is that, as you can see here, the attention 1 condition, which is attend to grading, has bigger difference than attention 2 condition, which was ignore the grading, in V4 than V1. And this is because V4 is an important brain area for attention. So therefore, this data confirms that phenomenon. So as I said, this is ongoing work. So we're planning to continue the spike analysis. We like to see the interactions of each factor, contrast, orientation, and attention within layers. So we like to see how these factors can influence the firing rate at different layers, and that will inform us how the layers communicate with each other, but also between factors. So there is an interaction between the contrast and the attention. Also, given that we collected quite a few sessions from the monkeys, we like to see the learning effect, meaning that there is an adaptation or learning in terms of firing rate because of those factors. But also we like to calculate specific features such as response onset, peak response, and peak response onset in order to run some stats and quantify the difference between the factors or interactions. At the same time, we're going to resume the LFP analysis. So we have an LFP data. So I forgot to mention that the LFP is the slow component of neural activity, whereas the spikes are the fast component. So the plan is to resume the LFP analysis. So we can run the computational spectral analysis of LFPs, but also to examine the effect of any interaction among the three factors on gamma and high gamma frequency band. So this what I have been doing since I came at the University of Nicosia Medical School. So the last part of the presentation is about the motor neuroscience, which that's my main focus of research. So here I will show you, I will introduce you science, knowledge that was derived, started at Medical University of South Carolina, where I did my PhD, and the New York University School of Medicine, my second postdoc. And the goal is to, by using that knowledge, to apply, to run a retrospective analysis about probing the descending motor pathways with TMS. So all of us, we know that the left brain controls the right muscles and the right brain controls the left muscles. And the reason of that is because of descending pathway, which is the cortical spinal tract. This tract is a fast monosynaptic. So only humans have the capacity to do these individuated movements and also fast. And the reason is because the brain has a direct access to the alpha motor neurons located in the spinal cord and immediately can innervate the contralateral muscles. Also, and this tract is dominated in distal muscles, meaning, for example, in terms of the upper extremity, corticospinal tract is very prominent in finger muscles versus the upper extremity muscles, such as the biceps, because the fingers are motor skill muscles. Whereas also there is another descending pathway, which is called the cortico-reticular spinal tract, CREST, which this tract has two main differences with CST. It runs mainly ipsilateral, but also is oligosynaptic, meaning it has more stabs before it innervates a muscle. Now, why we are interested in these two tracks? It's very important to understand the reorganization of those tracks, especially after a stroke. So now we have a lesion on the left hemisphere. Now the left hemisphere is what we call the ipsilesional hemisphere, whereas the right hemisphere is the contralesional, the good hemisphere. Now, the corticospinal tract that used to innervate the right muscles, now the right muscles are the paretic muscles, are the affected, are the butt muscles. And the reason is mainly because of the loss of function of the corticospinal tract. Now, at the same time, there is some evidence, especially from animal work, that the cortico-reticular tract, CREST, because it comes from the good hemisphere, may take over the innervation of muscles after stroke, which that means that what kind of promotes movement after stroke. So understanding this organization can help us to understand how recovery can occur after stroke. And if we know that, we can probe these pathways in order to promote recovery after stroke. So we can probe those pathways by using TMS, which is transcranial magnetic stimulation, which is basically, it's a coil connected to electric capacitor. When this capacitor is discharged, there is electric current that goes through the coil, then magnetic field, and then if the magnetic field is close to the conductive tissue, such as the brain, can create electrical current, which that electrical current elicits action potential, and then we are able to get what we call the motor evoked potential on the contralateral muscles. So by doing nothing and by stimulating specific parts of the brain that we know that are responsible for specific muscles. And as you can see here, by using neuronavigator, meaning brain GPS, we can accurately stimulate the specific location of the muscle. This is work done at the Medical University of USC during my postdoctoral work. So the main outcome of that stimulation is the contralateral motor potential, because this machine basically probes the corticospinal. So lastly, so now that I show you partial of my research work, my long-term goal, which hopefully you will be able to understand it while I was presenting, is to progress and develop mechanism-driven behavioral, computational, and neurophysiological strategies to optimize motor performance and recovery after neurological injury. Of course, this done, I show you, it could have been done without my supervisors, lab mates, research assistants, the people who participate in studies, the funding agencies who supported my studies, the foundations which donated equipment, and also, of course, my collaborators. Thank you very much.